Okay, so we have covered meiosis, mitosis, and now we're going to talk about some of the alterations or accidents that can happen in meiosis in particular. And the structure of the actual chromosome. Um, One of the things that can happen during during meiosis is that the chromosomes don't separate in anaphase. So this is called a non-disjunction. Disjunction. That's an N. And this is when the chromosomes, or chromatids, either one, failure of the chromosomes or chromatids to separate normally in anaphase. So, because meiosis has two anaphase stages, this non-disjunction can either happen during meiosis 1 or in meiosis 2. If it happens in meiosis 1, all four gametes are going to have the wrong number of chromosomes. If it happens in meiosis 2, half of the gametes that are made will be abnormal. Okay. So we'll take a look at that. So if one of these abnormal gametes is fertilized, the zygote or will also have the wrong number of chromosomes, right? So let's take a look at how that works. So here's meiosis 1 and meiosis 2. So here's a cell that's undergoing reproduction. And we've got the homologous chromosomes. This is during anaphase 1. The chromosomes are being, remember, pulled to opposite sides of the cell, right? Here they've separated normally. Let's do a red one where all these, they should have separated different sides, but all four of these, the tetrads, have all moved to one side of the cell. Once that cell continues into anaphase 2, let's draw actually two cells here. So one of those is going to have too many chromosomes, right? So now let's do the blue one. Now the sister chromatids have separated of the blue. But now we have got too many reds, and here we have none, right? So when those gametes are made, each one of these gametes is going to have a blue, 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 and two red with no red, right? So all four of these gametes are wrong or abnormal. Normal. So this is a non-disjunction in meiosis 1. Should I write this? Non-disjunction in meiosis 1. Okay. We'll contrast that to a non-disjunction in meiosis 2, rather. Disjunction in meiosis 2. Okay. So here in anaphase 1, both separate like they should into the opposite sides. But now there's going to be a non-disjunction in anaphase 2, or in meiosis 2, of the red one. Okay, so the blue one is going to separate normally, like it should. But now the sister chromatids do not separate in one of these. Okay. So then when the gametes are made, these are normal. They have one copy of each chromosome, which they should. This one's missing one, and this one's got an extra one, so half of the gametes are abnormal. Okay. This can be either kind of not very serious as far as the health of the offspring, or it can be fatal. I mean, this can lead to non-viable fetus, like basically just dies before it's even born. or can be cause some kind of health problems. One of the ways that we can diagnose whether or not a non-disjunction has occurred is we just take a picture, basically, of the chromosomes, and we can look at them. So we would use a microscope. This is basically a, let's call this an ordered display of the magnified, let's say, images of the chromosomes. So we can look at them like on a computer screen. Chromosomes. And they will kind of cut and paste them and arrange them so that they're lined up with their homologous chromosome. So this will allow observing either the chromosome number. So if there's a non-disjunction, if there's too many, and the homologous pairs, and any change to their physical structure. Sometimes pieces of the chromosome will accidentally either get taken out or fall off. It's or put onto the wrong chromosome. So one of the examples of a non-disjunction causing a syndrome, it's called, the fancy name is trisomy 21, commonly known as Down syndrome. And this is the most common chromosome abnormality. What it boils down to is the person has 3 copies of chromosome number 21. Because of a non-disjunction that we just talked about a minute ago. The symptoms, there's a characteristic kind of facial features. And they tend to be shorter, let's say shorter stature, how tall they are. They can have heart defects. And some developmental disabilities. Developmental disabilities. Okay. And the incidence of this goes up quite a bit with the age of the parent, both mother and father. So, for example, if we drew a chart and we put the the incidence of the syndrome and the age, of the mother, let's say. So 20, 25, 30, 35, 40, and 45. The incidence is pretty low. It's about 0.05%. But as the age of the mother goes up to 40, the incidence rate doubles to about 1%. And if we go up to 45, it goes up even more. So this is 3%. So it goes up quite a bit. So we have 46 total chromosomes, right? And we talked about this. 44 of them are autosomes, and 2 of them are sex chromosomes. Talked about that in class. And these are named the X chromosome and the Y chromosome. In most cases, if there's a change, if there's, say, an increase or a decrease in the autosomes because of a non-disjunction, it can cause a miscarriage. A change in the sex chromosomes is not as serious. So if you're female, so this is standard. Female has 2 X chromosomes. Standard male has a Y chromosome. So it's the presence of the Y chromosome that causes maleness. Maleness. So if you don't, so if you have no Y chromosome, you become female. If you have a Y chromosome, you become Y. No Y chromosome, I should put down there. Okay. So, this is, like I said, what they say standard biologically. So let's look at some incidents here. So let's put the mom's gametes up here, and this is kind of hinting at Punnett squares that we'll be looking at coming up pretty soon. So mom has got an X chromosome in both of her gametes, right? She doesn't have a Y, so all her eggs will only have an X chromosome in them. Dads can have an X or a Y. Gamete or a Y. So this is the standard male and female would fit into these boxes here. Standard female, XX. Standard male, XY. You get the X chromosome from the mom, the Y chromosome from the dad. Here, this baby girl got an X from mom and an X from dad. So that's just a standard female. Okay. If there's a non-disjunction, so when the cell replicates the DNA, it's going to make 2 copies of the Y chromosomes. And normally they would separate. If they don't, if there's a non-disjunction, two Ys can end up in the gamete. And so this person here would, if that happens, get an X from mom and two copies of the Y chromosome. Okay. This is a standard appearing male. Nothing wrong. They just have an extra Y chromosome. If there's a non-disjunction on the X chromosome, either from mom or dad, you can end up with, this is a standard appearing female. I'm just going to abbreviate that. But it's female because there's no Y chromosome, right? It just has an extra X chromosome. If there's a non-disjunction, and say this person here ends up with an X chromosome, only one, and we put a 0 or an O, so they didn't get a second, they only have one X chromosome. This is a condition called Turner's syndrome. Still female, but missing an X chromosome. If the same thing happens here and the person, the baby, I guess, gets one Y chromosome and no X chromosome, it's not viable. That baby will not survive, or that fetus will not survive. If they get two X's and a Y, this is a condition called Klinefelter. Make sure I'm spelling that properly. Yes. Okay. So it'll be male, but it has an extra X chromosome. And there are some characteristics. So Klinefelter is non-disjunction in Klinefelter, a non-disjunction in the egg or the sperm formation. XYY, this can be a meiosis in the egg or sperm formation. I'm sorry, in the sperm formation only. And XXX is a meiosis, non-disjunction in the egg or sperm, and then same thing here, a non-disjunction in the egg or sperm. So only the XYY, this is a non-disjunction, and D in the sperm. Okay. Okay, some other things that can happen in chromosome alterations. One is called a deletion. This is where part of the chromosome gets removed somehow. So if we had a normally this happens during when the DNA is being duplicated, when the cell is going to reproduce, or it can happen when during meiosis, when they cross over, it might lose a segment of it. So this basically, let's say it loses this segment of it, so it's basically just going to be a bit shorter, and then that segment gets lost. You can also have a duplication, where a segment of the chromosome gets copied twice. And it gets inserted on the homologous chromosome. So let's say, and here's the homologous chromosome. That's going to get doubled up, so one's going to kind of lose a bit, and the other one is going to now have more. Okay. Another thing that can happen is called an inversion. And this is when the chromosome basically gets taken out and flipped the wrong way and put back in. Oops, chromosome removed, and then reinserted in the opposite direction. Opposite direction. So if we were to, let's say, hypothetically have a chromosome with one gene there and another gene there, we would flip that around, and it would end up, instead of going blue, red, green, it would go blue, green, red instead. So it's basically reversed. And one last one called a translocation. And this is where segments of two non-homologous chromosomes swap. So the genes will end up on the wrong chromosome. So here's one chromosome and a non-homologous chromosome, and if they accidentally cross over and swap, you'll end up with a combination of two incorrect chromosomes. So of these, the inversions are the least harmful. Because the genes are still there, they're just in the wrong direction. That's not that big of a deal. The translocation, this may or may not be harmful. Not be harmful. If we go back, a deletion is potentially, I'll move this out of the way. Very harmful. Because if that gene that got knocked out or cut out of there is important for survival, well, guess what? Dead. Dead, or it could be just cause some serious mental or physical problems. So, for example, there's I'll just use it on this one. Sorry. Go. Deletion, potentially harmful. An example of a disease caused by a deletion is it's called Cri-du-chat. Sorry, no, it's not crit. It's Cri-du-chat, which means the cats cry. And if you look up this on like YouTube, they've got videos of babies. The baby has this condition where when it cries, it sounds like a cat that's like kind of like screaming and whining a little bit. So that's French for the cat crying. There's another condition called chronic myelogenous leukemia, or CML. Leukemia is a type of blood cancer, so this is the most common type of blood cancer. So it affects the white blood cells. And what happens is there's a reciprocal translocation. Between chromosome number 22 and number 9. So again, these are non-homologous chromosomes that swap segments. And this disease is one example of how that can affect someone's health. So chromosome, it's chromosome alteration. Chromosome is not always bad. But if you end up with multiple copies of chromosome, it's a condition called polyploidy. This happens more commonly in plants because plants are able to reproduce with themselves, basically. They're kind of hermaphroditic. And so, for example, like we're we're diploid, right? We've got 2 copies of our chromosomes. The wheat that we use to make bread has got 6 copies of chromosomes, so it is, hexaploid. Copies. Strawberries are octaploid. Well, they can be. So they can have 8 copies. There are some frogs that are tetraploid. So some frogs. Tetraploid. So they have 4 copies. There are some oysters. Some oysters are triploid, so they have 3 copies. You kind of get the idea here, right? So there we go. That is the end of the cell reproduction chapter. And that's it.